Okay, so before we begin, today we're going to continue with our review of glucose metabolism, aka cellular respiration, aka glucose oxidation. So I want you to outline what you remember from last time in previous courses about what our parts of this process are, where they're happening in the cell, and how much ATP on net your cell is producing for each of those steps. All right, so I'll give you a couple minutes to do this before we continue on where we left off, which was with the Krebs cycle. So one of the things we're practicing here is a helpful way to study for people can be for processes to redraw them for yourselves. When you can consistently redraw something, that's often a sign that you got it down. So. Okay, take another minute or so there to finish up. And then I'll draw on the board with you. All right, let's go through it. Okay. So we're starting out with glucose. And so our first step that we went over was that first way that we begin to break down our glucose. So we're basically breaking it in half. Okay, so as we break down glucose, that first step is called glycolysis. Okay. Glycolysis anytime you see lysis, that ending means to like break something down. So that already has in its name what it's doing. Okay. So then we have a kind of intermediate step that at least in your book doesn't have its own specific like number, but we're going to put it on here anyway, just so we remember it happens. All right, so this is that pyruvate linking step. Okay. Okay. All right, pyruvate. And where we left off is with our next step, the Krebs cycle or the TCA cycle. And our final step will be pretty much focusing on today is oxidative phosphorylation using the electron transport chain. Cool. So those are the steps we're focusing on. And numbering them this way means that you know which ones are going to be producing ATP. This is why your book kind of doesn't focus on that pyruvate linking step, because we're not producing any ATP there. So for our counts for ATP, for glycolysis, on net, we get 2 ATP. We had to put in 2 to kind of prime the pump, got 4 made in the rest of the process. So our total glucose that our total ATP that we've kind of earned here is 2 ATP molecules. For our Krebs cycle, we'll get back to it and take another look at it real quick, but it's cycling twice for every glucose molecule. Every time it cycles, it produces 1 molecule of ATP. So this is actually also going to give us 2 ATP. We haven't gotten to the oxidative phosphorylation yet. And this is where we have some more variability in number. So for ATP, we're going to go with lots. Okay. So I'm not going to expect you to memorize an exact number for this, but it's an order of magnitude greater than either of those 2 earlier steps. We'll take a look at why that is today. Okay. So the other part of this question is we want to know where in the cell we are. Because this is going to be important for eventually when we talk about like, okay, does this cell have a mitochondrion in the first place? So here I'm going to draw my cell. And I'm going to give us a unrealistically large little kidney bean mitochondrion here, okay, with its inner membrane swirling around. So now I'm going to put on the numbers the way I numbered them over here on the left. Okay. So for number 1, glycolysis, we're out in this general part of the cell, right? So we're in the cytosol if we're thinking about it as a solution. Sometimes we're calling it the cytoplasm, depending exactly what we're focusing on. But we are not in a mitochondria yet. So we don't need mitochondria for glycolysis. Other things we don't need are like oxygen. Right? So you actually don't need all that many cellular components. You don't need those same conditions to run glycolysis as you do if you want to run this whole process of aerobic cellular respiration. So glycolysis is out here. And then we have our linking step with pyruvate. So that's going in all the way into the center of our mitochondria. So that's where our Krebs cycle is going to happen. So I'm going to put 2 here. And when we get to our oxidative phosphorylation step, that's also happening in the mitochondria. I'm going to put the 3 here, but as we'll see in a second, this step is really talking about how we're crossing this inner membrane. Right? So we're going to see that we have stuff flowing from this intermembrane space into the matrix of the mitochondria, so into the center, which is where you're going to end up with the ATP. So questions about this kind of big picture overview of the process. Okay, cool. You got this. Then you think, well, we'll be most of the way towards what you need to know, at least for our exam, but we are going through this in more detail. Okay, so when we left off, we were looking at that Krebs cycle. So we know that it's a regenerative cycle where we have it looping around and around and around. Its starter molecule being reproduced at the end of the process. We can reuse kind of all these enzymes that it has in its intermediate steps. What we see here are the details of exactly how each of those individual steps is happening. We're not focusing on memorizing them individually, but you can see there are 8 of them. And what we're doing is basically peeling off parts of these molecules. We're getting a lot of reduced electron carriers along the way. All right, so I'm circling those, squaring, I guess, those reduced electron carriers that are going to be carrying electrons to our final step. Okay. And we are getting rid of the carbon that was in our glucose molecule. So we're breathing off CO2 here. Okay. And we're making, for each turn of this cycle, 1 molecule of ATP. So you can see here that we're starting with this ADP molecule kind of floating out to the side. So that's the adenosine diphosphate, so just the 2 phosphate groups. And in this process, as we're doing step 5, we're releasing some energy and getting it stuck in this bond between that final phosphate group of the ADP and this additional phosphate group that we're adding on. So this is how we get that 1 ATP per cycle of the Krebs cycle, but we go around twice for each molecule of glucose because we broke glucose into those 2 pyruvate molecules during glycolysis. So this is enzyme mediated. You can see this here. This was substrate level phosphorylation of our ATP production. This is the same kind of phosphorylation, so the same sort of general way of adding a phosphate group as we see in glycolysis. All right. So as we get into our step, this is going to work pretty differently than glycolysis and the Krebs cycle. Okay. So for our Krebs cycle and glycolysis, up until this point, if you add the 2 together, on net, we've made 4 ATP molecules by the time we get to this process, 2 from glycolysis, 2 from the Krebs cycle for every molecule of glucose. Okay. Aerobic cellular respiration makes a lot more ATP than that, right? So we're ballparking somewhere in the 30-ish range, okay? Most of that ATP is being produced by the electron transport chain through this process of oxidative phosphorylation. Now, the way oxidative phosphorylation works is pretty different because instead of having these like numbered and enzymatically mediated steps, right? Like all those different named enzymes in the Krebs cycle and the named enzymes that we didn't bother naming in glycolysis. Now we're kind of passing electrons down a series of membrane proteins stuck in the inner membrane of the mitochondria. So it looks different, it works different than those earlier parts of this process. So we're using this movement of electrons really to kind of fuel our ATP production in this oxidative phosphorylation step. Another really important distinction about this oxidative phosphorylation is that it requires oxygen. Actually, we haven't used any oxygen at all until we get to this oxidative phosphorylation step at the end of our electron transport chain. Right? No oxygen in glycolysis. We didn't need to use any oxygen to move pyruvate into the mitochondria. We didn't need any oxygen in order to do the Krebs cycle. We don't need any oxygen until we get here. So it's really this process that's why you need to breathe oxygen, right? Literally just right here on the inside of the mitochondria at the very end of this step. But it's super important because this is where you get so much ATP, right? You need that ATP, that energy to live. So the electron transport chain itself is a chain of molecules embedded in that inner mitochondrial membrane. So I was drawing that mitochondrion before, right? So outer membrane here. So outer. Okay. Sorry about my atrocious handwriting. Inner membrane is here. Okay. And we've got some spaces in that mitochondrion as well, right? So there's a space here, a physical separation between the inner membrane and that outer membrane. So that's our inter. So we're not going to confuse this with inner. It's inter for between. So this is the intermembrane space. And then our other sort of conceptual space that we're talking about is this area enclosed by the inner membrane. And so that's the mitochondrial matrix. So those are our locations in the mitochondria. This is like basically the anatomy of a mitochondria. So the electron transport chain, when we're saying that we have a chain of molecules in that inner mitochondrial membrane, we mean we've got stuff embedded here in that membrane. So we've got these molecules going across that inner membrane. So it's going to be happening as we go down the electron transport chain when we talk about it, right? What's happening is a series of oxidation and reduction reactions, so like redox reactions. We're passing these electrons down the chain and we're going from high energy to low energy. And what we'll see is that as we do that, what we end up doing is like using that energy to create a buildup of hydrogen ions here in the intermembrane space. Okay. And then when we release those hydrogen ions and let them spread out the way that they want to, we're going to get hydrogen ions in this final step flowing into the matrix and they're released. And basically what this is we're releasing like potential energy, like that tension of there being lots of hydrogen ions in one place and none in another place. And we use that movement to create ATP. We use it as energy, just like you use a windmill to produce energy on a wind farm. That's how it's working. Okay. So we're releasing this potential energy held in this gradient to synthesize ATP, which is different than an enzyme like picking stuff together, putting it together, pulling it apart, which is why we have a separate name. So this is the oxidative phosphorylation step. That is where we're going to be using our oxygen right at the end of the electron transport chain as we're getting ready to release this hydrogen ion gradient. There is a protein that those hydrogen ions are going through. So that final molecule is called ATP synthase. So it is an enzyme, but it works differently than enzymes we've thought about before. Its job is to synthesize ATP, right? So synth is for synthesizing. A is telling you it's an enzyme. ATP is telling you what it's doing is making ATP. In this process, we're going to need a final electron acceptor as we're going through the series of passing electrons down and essentially taking more and more energy from them. And that final electron acceptor that basically gets like our leftovers is oxygen. So that's why we need oxygen. Because otherwise, the whole thing gets kind of backed up and jammed, and you can't do it. Okay. So we have this donation of electrons to the electron transport chain. And this is why NAD and NADH were important earlier in the process. So those were our electron carriers. So they're essentially kind of like carrying electrons in this little bond here, basically, right? So they're carrying electrons from earlier parts of the process, right? So we made some NADH in glycolysis. We made some, a little bit in the pyruvate linking step, but we made a lot in the Krebs cycle, right? That was the place where we made most of our reduced electron carriers, right? So they're carrying these electrons to the electron transport chain. And that's where we're getting the electrons for energy to make this work, right? So the electrons. But this is also where those hydrogen ions are coming from, right? So we're building up a gradient of hydrogen ions, right? So we like peeled off the hydrogen ion, got the energy of the electron, and we moved to the hydrogen ion kind of in the process. And this works for both our types of electron carriers, NADH being the more common one, but FADH2 is produced in the Krebs cycle in limited amounts. It works the same. At the end of this process, we need to kind of like tie it all up with a bow. So our final accepting of the electrons at the very end of the chain, we take them, put them back together with some hydrogen and a little bit of oxygen and get water, which is why water is one of our products of cellular respiration. So we need to put oxygen in so that we can kind of finally finish off this process and get water as our byproduct and be done with everything. So you can see that happen here and better drawings than mine. Okay, so what we're looking at, we're zoomed in here on that inner membrane, right? So we're inside a mitochondrion. So here on the bottom, this is where our mitochondrial matrix is. Up top here is our intermembrane space. What we're looking at are those molecules in that inner membrane that are passing the electrons. So we can see here coming up to our first molecule, we have NADH and some hydrogen ions. We're peeling off electrons and hydrogen ions, and we are forcing hydrogen ions through into that intermembrane space. Okay, so the hydrogen ions in this image are the blue dots. Okay. So what we're doing is we're building up more and more and more concentration of hydrogen ions, which are basically just individual protons, in this intermembrane space. And we have to use energy for this because we're fighting a gradient, right? Stuff doesn't want to be jammed together, just like you don't want to be jammed onto a like super crowded bus, right? So we need to use some energy to push everything together. And that's what we're doing with those electrons. Okay. So we're going through, we're bringing in our electron carrier, we're pulling off hydrogen ions, pushing them into that intermembrane space. And then in the mitochondrial matrix, we're going to end up left with NAD+, right? So we peeled off the hydrogen ion, we peeled off an electron. We've got this molecule, this precursor molecule that we kind of started with at the beginning, back. We do it again and again, down the chain, kind of passing the electron. At the end, we're left with an electron, kind of low energy here, some hydrogen ions. We combine them with oxygen, get water as our byproduct. So this is us releasing kind of the end of this process so that we can keep using those enzymes. And this is what allows us to use ATP synthase, this enzyme at the end, to release the hydrogen ion gradient. So this is kind of paired at the end here. So what's happening is that we have all those hydrogen ions now allowed to flow into the mitochondrial matrix. And that movement is energy that we use to tack on an extra phosphate to ADP. Bless you. Getting ATP, our unit of cellular energy. So over all the process of our aerobic cellular respiration, aerobic because we use oxygen, think you need air for it. Okay. We start with glycolysis, where we get our 2-net ATP. And we can see now the importance of the fact that we're also producing this NADH, right? So we put in some NAD, reduced it, made some NADH here in glycolysis. We send those all the way into the mitochondrion, just like we send in pyruvate. So here's that pyruvate linking step. We're turning it into this acetyl-CoA. We've got 2 of them because we split glucose in half at the beginning. In this step, we are producing some of that reduced electron carrier as well. We're peeling off some carbon dioxide from the glucose molecule, breathing off some CO2. These reduced electron carriers are also going into the mitochondrial matrix so that they're there to power this electron transport chain. Krebs cycle, same deal. We make a little bit of ATP, but our main kind of point of this process, what's going to end up doing a lot of work, is that we're making a lot of reduced electron carriers here, right? So we're totally making 6 molecules of NADH here versus only like 2 in each of these earlier processes. Plus, we're making our second type of reduced electron carrier. So we're putting a lot of energy into these electrons that we're sending to the end of the process. At the end, we take all of those reduced electron carriers from all the places that we've made them. They've been transported into the mitochondrial matrix, so they're in the center. And then we're using their electrons in order to build up this hydrogen ion gradient. And then at the very end, once we've gotten all the energy out of the electrons, we have to tack them onto an oxygen, which is why you end up producing some water at the end of the process. And all that energy that we've been using, or all that energy that we've been storing in those reduced electron carriers, is powering the electron transport chain in oxidative phosphorylation, producing a lot of ATP, somewhere around 30-ish molecules of ATP per molecule of glucose. So this is the whole process. This is the basic fundamental way that you are usually in most of your cells breaking down a glucose molecule. So this is what you're doing anytime you eat a carb, right? Glucose is a carb, right? This is how you're getting energy from your food and turning it into a type of energy that the cell knows how to use, that the cell is able to use. But we can only do this if we've got a mitochondrion, right? Because this happens at the end. This final part is happening in the mitochondria. And we can only do this if we have oxygen, right? You don't have oxygen, and we can't tie this process off at the end. Everything backs up. The electron transport chain gets backed up. We're not turning all those NADH molecules back into our NAD+, which means we're running out of them. So we can't keep feeding like the Krebs cycle here, right? If we don't do something else to get them back, we wouldn't even be able to feed glycolysis, even though that's not happening in the mitochondria at all, right? So this whole process would get jammed if we don't have oxygen. Kind of like a paper jam. Okay. So this is a full equation with slightly different numbers for the ATP, which is why we're not focusing on them too hard, different in different tissue types, even different for like people of different ages. So this varies a lot due to kind of individual characteristics of your biology. So here, oh, I thought blue now. Did those questions last time. I just want one kind of vocab question because I'm about to use a word. Okay. So when I say we're talking about a catabolic reaction, what does that mean to you? take another second or so. And let's take a look. Okay, cool. Yes. So when we describe catabolic reactions, we're talking about ones that are breaking down a molecule. So the opposite of catabolic is anabolic. So when we talk about anabolic reactions, that would be building something up bigger. Okay, so these are working in opposite directions. I personally always remember this in a very kind of silly way. I just pretend I have a very tall friend named Anna with a very tiny cat. And I literally picture this in my head every time I see this word at this point. Just silly insight into my brain. Okay, so catabolic reactions. What we've been doing, we've been breaking down glucose, right? So this is a catabolic process. We started with a larger molecule, we're getting smaller molecules. Okay. The reason I was asking you that is because next what we're talking about how we could break down glucose if we don't have any oxygen, right? So when we have glucose catabolism in the absence of oxygen, that's still possible, right? So this happens for weird organisms in weird situations, but it also happens in your own body, right? So you might have low oxygen availability for kind of a number of different reasons, right? Like it might be the case that you're in a room that's rapidly losing oxygen for some reason, but more often we're thinking about like you, right? We're thinking about you're a muscle cell, right? Your muscle is working really hard. Oxygen was coming in through the blood, but you're working so hard that you've used up all the oxygen available to you. Blood flow isn't keeping up. Maybe the muscle is getting so big that the blood flow is getting cut off, right? So your individual cell can be operating in a situation where they don't have all that much oxygen available. And often if they couldn't do anything else, is they could just die, right? But you're not totally out of luck if you're in this low oxygen situation from a cellular perspective. You can still make some energy, right? Because we didn't need oxygen for every step of that glucose breakdown, right? We needed it to end the electron transport chain at the very end to do that big boom of producing lots of ATP. But we didn't need it earlier on. We didn't need it specifically, what's going to be important for this part, we didn't need it in glycolysis, which was happening out in the cytosol, in the cytoplasm. Okay? So if we have low oxygen availability, that electron transport chain is going to get backed up. The Krebs cycle is also there in the mitochondrial matrix. It's maybe running out of NAD+, right? It's going to slow down and stop. It's getting jammed up too. But all of that is happening in the mitochondria. Outside the mitochondria, where glycolysis is happening, you still have glycolytic enzymes. You still got some stuff that you can work with. So glycolysis can continue if you don't have oxygen. So you could still get those 2 ATP molecules out of your glucose. It's not a lot, but it's something, right? So you could still get those 2 net ATP from glycolysis. But you can only do that if you take the NADH, that reduced electron carrier that you've been making, and you put it back into its original form, that NAD+, so that you can keep reusing it, right? Otherwise, it's also going to get backed up. It's going to run out of NAD+, and this process isn't going to work either. Okay. So this is where we talk about like lactate or lactic acid, although told by the physiologists that they really want you to say lactate, right? More technically correct. Exactly what the details are on that, I'm not totally sure. So lactate, we're going to be making lactate. And the point of making lactate is so we can continue to do glycolysis over and over again and continue to get a little bit of energy for our cells, even though we don't have enough oxygen present. Okay. So here's what that looks like, right? So glycolysis works the same way, right? So you're putting in glucose, right? You're getting some ATP, 2 on net, so 2 total you're earning. And along the way, you were producing some reduced electron carriers, right? So our arrows are going this way for the reduced electron carriers. In order to do that, you were putting in NAD+. All right. So unfortunately, you can't just run glycolysis and cross all of this out, right? Like that's not how the enzymes work. They need to do that part, even if there's nowhere for those reduced electron carriers to go, even if there's no electron transport chain running. We can't just like not do one of the 10 numbered steps of glycolysis. Okay. So we need a different way of getting those reduced electron carriers oxidized back to their original form. Okay. So that's where the lactate comes in. So the end product of glycolysis, in terms of what's going on with our big carbohydrate, so we've turned glucose into a pyruvate molecule. Okay. So we got pyruvate. So we've got pyruvate, we've got 2 molecules of ATP, we've got these reduced electron carriers. And what we want is we are trying to get that NAD+ back so that we have enough to keep doing glycolysis over and over again. So the way we do it is we run another separate kind of side process. So instead of taking that pyruvate and moving it into our mitochondria and the linking step going into the Krebs cycle, right? Think about it as like our mitochondria is broken. We don't want to do any of that. So we're going to use that pyruvate to regenerate that NAD+, right? So what we do here is we take our pyruvate, this enzyme lactate dehydrogenase produces our molecule lactate, right? So we turned the pyruvate into lactate. And a convenient aspect of this process for us is that in order to do that, we needed some NADH, and it got turned back into NAD+. So this is fundamentally kind of like why we're making the lactate, is it's to deal with this kind of electron carrier problem that we have. So when you talk about like lactic acid buildup in a muscle and saying like maybe that's why your muscle is sore, what we're talking about is your muscle was working hard, it ran out of oxygen, it still wants energy because it's still working, it did glycolysis, started running out of this reduced electron carrier, started overflowing with this reduced electron carrier and not enough of this starter molecule. So it turned pyruvate into lactate, so it could just keep cycling back through glycolysis over and over again, so that you keep having like a little bit of energy. So this is what happens when we're in an anaerobic condition, right? This is analogous to the way that like alcohol is produced in fermentation, right? That's kind of dealing with the same situation where we want to keep getting energy, but we can't run all that powerful electron transport chain stuff. Okay. So, but the glycolysis is just the same old glycolysis as our normal aerobic cellular respiration. So there are basically like 2 conditions in which you're going to see glycolysis. We got kind of like different end results here. Questions about this kind of comparison between what's going on when we've got oxygen and when we don't have oxygen? Cool. All right. You guys have this link. This is just a little video from your textbook showing you in quote unquote 3D of what this process looks like. But I want to make sure that we go through and talk a little bit about the fact that we can break down things that aren't glucose, right? Because we're not just eating sugar pills all day long. There's a point to that too. Okay. So our immediate, easiest to break down from a nutritional perspective form of energy are carbohydrates, particularly glucose. Really easy for your body to process. We've got this glucose metabolism, this aerobic cellular respiration. It's efficient at breaking down glucose molecules. And often that's where we kind of stop when we're thinking about how ATP is being produced, how metabolism in your body works. But we do have other places energy are stored, in particular lipids, right? Like fat is a lipid, right? Triglycerides are oils and fats, right? Like butter and stuff, or literally your fat cells have a lot of lipid in them. They've got energy in a more like kind of long-term storage form. So we want to be able to get energy out of those when we need it. Your body can do all sorts of creative things. So it can actually also break down proteins. You don't necessarily want your body to start eating its own muscles, but it can if you're in a kind of like famine situation. So we're going to take a brief look at how those processes work. They're not going to memorize any new cycles. We're just going to kind of look at how these processes compare. Right. So you can see in the center here, actually the same process, right? So here in the center, we have our typical aerobic cellular respiration, breaking down glucose. Okay. Cool. So that's our normal, that's our baseline. If we're working with glucose, if we're working with carbs, we can mostly do this. Okay. If we're working with proteins or fats, particularly triglycerides, what we're going to do is kind of use parts of that process. So we're going to kind of come in from the side, get some of these intermediate molecules from other places and other processes, and then continue to hopefully even run the electron transport chain at the end and do oxidative phosphorylation. So over here, let's start on the left because we read left to right. So the proteins, you can see here, we would break proteins down into their amino acids, right? A protein is just a big folded chain of amino acids. Okay. We can take those proteins, and some of them we can use to make acetyl-CoA, right? So we can make acetyl-CoA from proteins and amino acids. Okay. We're not going to think about any of the particular details of that, but there are processes that allow you to do that. If we have acetyl-CoA, the Krebs cycle doesn't care where they came from. So if we have acetyl-CoA that came from an amino acid, super. We can still go through the Krebs cycle all the way to oxidative phosphorylation, all the way to that big bang ATP production at the end in your mitochondria. That works great. If you have a triglyceride, so a fat, that was our one that looks kind of like a flag. It had that glycerol backbone, and then it had our fatty acid chains on the end, so this big long hydrocarbons. Okay. We're going to process these two parts in different ways. Okay. So from the glycerol piece, okay, so that glycerol piece, also is like an alcohol, it works like a carbohydrate. That glycerol, we can put into glycolysis into some of those intermediate steps. We've got kind of sideways processes that'll help jump into glycolysis. And so from that glycerol, we can actually pretty much run our whole usual aerobic cellular respiration. Not necessarily as effective as if we had our glucose molecule, but pretty much the same. From the fatty acids, we could produce some acetyl-CoA, and we can jump kind of even farther down into the process to right before we run the electron transport chain and have kind of alternate ways of getting that electron transport chain to run and doing oxidative phosphorylation. So we like borrow parts of this big full flow that we go through when we talk about glucose metabolism for breaking down other types of molecules. Another thing you can see on here that we will be thinking about in more detail in the future is that glucose has a storage molecule, right? So if you link a bunch of glucose together into a polysaccharide, in an animal cell, linking glucose together gets you glycogen. So glycogen is lots and lots of glucose together. If you were a plant, linking a bunch of glucose together would have a different shape. That polysaccharide would be a starch. Okay? So from that glucose and glycogen, we have a relationship where you can take glycogen, peel off those individual glucose molecules through a process called glycogenolysis, right? So breaking down that glycogen. And if you have extra glucose, you can actually store it in the glycogen. That's the point of it. So you can do glycogenesis as well. So these are all kind of like spin-offs of this main idea. So for the glycogen metabolism, you're this is happening in mainly your liver, but also a little bit in your kidney. So you've got these glycogen molecules where we've stored glucose. And in that case, when we don't have enough glucose because you're like not eating a donut right now, you can take that glycogen stored in the liver and the kidney, peel it apart, and provide glucose to your other cells. And in particular, we're going to see that your brain and your nervous system, they don't really have the machinery to like break down proteins and fats and stuff. They kind of need just glucose, right? They've just got this simple basic process available to them. So you would want to take the liver and break out those glucose from the glycogen to feed your brain under these conditions. In this glycogen metabolism, the glycogen is inside a liver cell. But if you're trying to get all the way to the brain, right, like you need to send something through your bloodstream all the way up to your brain. And glycogen itself, like, can't really leave the cell. So you have to break off the glucose first. And we have an intermediate kind of molecule that gets made in the process of breaking down glycogen. Okay. So as we break down glycogen, what we're going to be doing is pulling out these little glucose units, and then we're using the enzyme glucose 6-phosphatase to convert like the glucose, but like plus it's a little kind of connector bit. That's our glucose 6-phosphate into just normal old glucose that can leave the cell and get into your bloodstream. So it's kind of like a three-piece thing, right? You start with glycogen. As you try to pull off these glucose units, you get glucose 6-phosphate. It's not shaped right to leave the cell. In the liver and the kidney, you've got these enzymes that turn it into normal glucose. Now it can leave the cell that it's in and get into other cells because your body has normal ways of dealing with normal glucose. Okay. We can also make brand new glucose, right? So not glucose that we stored in glycogen, but glucose that we made like from other components that we're getting from random other processes in your cell. So you can actually make glucose from that glycerol molecule holding your triglycerides together. You can make glucose from lactate, actually. You can even make glucose from an amino acid. We have enzymes that do those processes. Okay. So when we do that, we call it gluco for glucose, neo for new, and genesis for creation. And making new glucose is gluconeogenesis. And I am sorry that sounds so much like glycogenolysis, which is breaking down glycogen, but those are just the two terms. So practice comparing how to look at a word and know you're breaking down glycogen versus this word where you have that neo in the middle, you're making new glucose. Okay. So this is also mostly happening in the liver. And this is not exactly the reverse of glycolysis. There are other enzymes involved, but you can think of it a little bit like running some of this stuff backwards. Okay. So our end goal is to make our glucose. So one way we were making glucose was from breaking down glycogen, pulling off these subunits, getting glucose 6-phosphate, turning them into normal glucose. Another way that we can do it is pulling together these components with different enzymes and doing kind of the same thing. Okay. Cool. So the whole process of like relating these terms to each other is here. So this is something you should practice. Right. So glycogen, if you want to break it down, glycogenolysis, giving you glucose 6-phosphate, which you can turn into glucose with an enzyme that leaves the cell. If you make glycogen, you'd be going from glucose all the way, tacking it all together into big long molecule. And we also have a separate process of gluconeogenesis that makes glucose. Okay, so I can see we're at time. So we will stop here. So I have something that came up. So we won't have in-person class on Friday. I'm going to put up a video and an assignment on review of the cell membrane for going into our next topic. So I'll post that and the details of how to complete it, but that's what you should be doing with your normal class time.